So today I'm going to give the second part of the talk, and basically last time, just to remind you, we were talking about the dynamics of sensing and growing system, right? Such as inspired by plant tropisms, so mainly shoots or roots. But we completely disregarded the effects of growth, right? We were assuming all the time, because we're looking at a minimal model, so we're assuming that L, the length of our organ, is constant, and we're able to extract a lot of information about it, right? We're able, in particular, to show how the steady state shape can tell us something about the dynamics while reaching the steady state. So that was pretty cool. But now I want to see what we can say about the influence of growth, and also just, I mean, not just the influence of growth, but how to connect the equations that we saw before, which were based on geometry at the end of the day, with growth. Okay, so let's see how we go about it. So I'll show you again the movie that I showed last time of the wheat coloptile. And if you can see, there are these dots on it. So these are just markers for us so that we will be able, with image analysis, to follow growth. Okay, so what I marked here, this arrow is between like this dot and this dot. Okay, so try to follow them with time. Okay, so to make things easy, I mark them with another arrow. So you can see how this particular segment, which I randomly chose in the middle of the wheat coloptile, got elongated dramatically. I'd say it's maybe 3 times longer than it was originally. Okay, so if before we were considering all the dynamics of this movement, we have to remember, I mean, I think this really drives the point home that growth is not only the driver of these movements, but it also has a very big effect on the morphology that we're seeing. And more so, maybe we can, I'll touch this again later, but okay, this segment was somewhere in the middle, right? And it's elongated to this size. But this also means that it pushes upwards all of the material segments up here, right? So basically you have, imagine a lot of people, one next to each other, and we're all expanding at the same time, so we're all pushing each other sideways. So you have to somehow account for all of this. And then if you have to account for this, the question is, how do you regulate? How is it that even though each segment is elongating so much, and they're all pushing each other by so much, how is it still you get these nice and smooth dynamics? Okay, so this will be the question that we will try to tackle today. So how can growth induce curvature? So it was touched a bit in the previous lecture before lunch. Basically, imagine that you have, this is a segment, a part of your organ. And now you have 2 sides. One side elongates with a growth rate epsilon1 dot. Okay, dot means that it's a rate. And the second one with a different elongation rate, epsilon2. Let's say this epsilon2 is a higher growth rate compared to epsilon1. And after some delta t, you'll get this curved form, right? Because the outer part is growing faster than the inner part. So just a word about how we define elongation, the elongation rate. If the elongation rate is 0, it means that there's no change. Okay? If it's smaller than 0, that means that you're shrinking, but we do not consider shrinking in these models. And if it's larger than 0, then you're stretching. Okay? This, we'll be using this further on. Okay. So now I want to start with the idea here is now that we want to show, now that we know that this is the way that we get curving from curvature from differential, from 2 specific growth rates, epsilon1 and epsilon2, right? We know that in our equations we're continuously talking about kappa, the curvature, and the change of curvature in time. So now the point will be to try to relate this change in curvature with actual growth rates. And this is what we're going to do here. So the first thing to do is to get some geometrical relation. We'll see exactly what. So if this is our segment, okay, and we say that the radius is twice r, okay, r is the size of each side. Delta s is the length of the segment along the median, right? Because remember that our parameter s goes along the median of the organ. And if we consider, right, this has a certain curvature, kappa. So the radius of curvature, that's basically, if I were, when I have curvature, I can define the radius of curvature. If I were to complete this as a circle, then the radius is a radius of curvature. And it's, you find it like so. R is 1 over kappa, okay, from the median. And the angle here is some theta, okay? So now I can say, okay, I have the inner part and the outer part. Delta S2 is the inner part, the shorter part of the organ, and delta S1 is the outer part. And I have for each of these also the radius of the curvature. Okay, so R, I misplaced them, they're the other way around. R2 is the shorter radius, and R1 is the bigger radius. And at the end of the day, out of a lot of shuffling with the geometry, what you get is this ratio, okay? So here I relate my curvature kappa to the lengths of the segment, of the outer part and the inner part. And you can already guess that the relative length of the two segments is associated with the differential growth rate, right? So originally I was thinking of showing you this calculation, but it's just geometry. So in the interest of time, I think you can believe me or read the paper, and we'll continue from here. Okay, so now let's see. We have a small segment of the organ, and we know, as in the initial picture, that we have two different growth rates that will give us this form here. How do we get the change in curvature over time? Okay, so we say after some time dt, okay, this time between here and here, this side is elongated a bit, this side is elongated a bit, and the curvature has changed a bit, right? So we say that the new element size for delta S1 and delta S2 becomes this thing, right? Because remember that epsilon dot is a stretch here. So you have to multiply, so it's a rate, so you multiply it by dt, and you multiply it by the original length. So these are the new lengths after dt, and the curvature will also be kappa with some differential, a new kappa, okay? So now if I substitute these into this, and I mess things around, at the end of the day, I get this equation. So this equation relates, basically gives me the derivative of kappa in time, how my curvature changes with time, as a function of the two growth rates. So let's do this calculation. Yes, okay. So we have the original equation. The geometrical relation was kappa r equals, okay. Tell me if I'm wrong because I don't have it written here. ds1 minus delta s2 divided by delta s1 plus delta s2, right? And we said that we have that delta s1 goes to delta s1 plus delta s1 epsilon 1 dot dt. Okay, so let's substitute this in this equation. Okay, now I want to reorder this, but putting together all of the orders, what we care about, right, are these the differentials, small elements. So I want to reorder everything in this way. So we get, again, kappa r plus of course, I know. Okay. Okay. So actually, this is a very long calculation now that I look at it. Trying to think if I can shorten it a bit. Okay, I'll do a few steps and I'll jump in between, okay, to get you from one point to the other. So here I'll do one more reordering phase. So this side, okay. So here what we can do, right, I have a constant something multiplied by dt, and then I have this messy thing. So the idea is that if I have 1 over something small, right, because this is multiplied by dt and dt is small, then I can I can make an approximation 1 over u, when u goes to 0, this is 1 minus u, right? So this helps me linearize this effect, this term. So I'm going to use it here. Now let's see where I can go to. Yes. Okay, so when I do this, I will have basically, instead of 1 over, I'll have 1 minus dt multiplied with this. And when I open up here the parenthesis, basically I get a multiplication of this with what I have here, but I have at some point dt squared when I multiply by this. So this drops because we're only looking at first orders, right? So from this we get 3 dots I'm putting here. Delta S, so this equals delta S1 minus delta S2, delta S1 plus delta S2, plus dt of epsilon dot 1 delta S1 Okay. Now this, you remember, this equals kappa r, right? So already see that this is going to drop with this. Okay? And now there are a few other, if you open up everything and you fix things, at the end of the day, what you have is, so d kappa, okay, I'm starting from that side, equals, where shall we, 1 second. Okay, at the end of the day, you have d kappa r equals, I won't show the whole calculation, just the idea, is equal to dt multiplied by something. Then when you divide by dt, you have your derivative in time. So this is how you get the derivative in time. As for the epsilons, yes, the epsilons, of course, you get here. So you get this just by reordering. But this is the idea, okay? So by taking the differentials in for delta S1 and delta S2, and they are a function of dt, of time, and then taking d kappa, that's how you get your derivative at the end of the day, which equals what you see here. Okay, so this is really too long for, I should have thought about this. Yeah? Yes, you're assuming that this is a small, okay, I should say that this calculation is not true for the whole organ. Okay, this is local for a specific segment. So we're assuming also that you have a constant curvature throughout the segment, right? Because in the whole organ you have the curvature changes with the place. So notice that here I have just a derivative of there's no dependence on S here, okay? It's just a specific segment, okay? Just a small element in the material. Now I want to go and incorporate this in the whole organ. True. So no, here we're taking the simplest case. Okay, so now I want to put this as part of, imagine that each element acts like this, but we want to see how the dynamics of the whole organ looks like. So to do this, we have to take into consideration a bit, basically it was what I showed you at the beginning, that if you look at the distance between 2 points, and you see how it grows and pushes everything away. So this is a more involved or more detailed experiment. It's a classic Sachs experiment, because Julius Sachs was the first who did this many years ago. So I'm going to show you a movie on the left, and on the right what you see is snapshots of this growing root. And what they did is they marked with dots equally once every millimeter, okay, along the root, and they see what happens to the dot. Here you see it in live action, but what you see here is that, so the growth in this area is obviously 0. Nothing has changed. Okay, so the red arrows here tell me where the position of the black dots are at the beginning, and here the red arrows tell me where the position of the same dot is at the end. Okay, so you see that at the beginning, the positions of these two, I mean the parts which are farther away from the root tip are identical, so there's no growth. And as you go further down the root, there is more and more distance that's covered due to growth. So what this tells us is that there's an area here, right, which is associated with this area, subapical area, where the growth rate is maximal. Okay, but it's not necessarily homogeneous. But the point here also, from the mathematical point of view, is that there's a difference between talking about a specific a material element. Okay, so I'm looking at cell number 10. Where did cell number 10 go? Right, cell number 10 will always be between cell number 9 and number 11. Okay, so that doesn't change with time. But what does change with time, it's its position in space. So here's where we resort to Lagrangian and Eulerian coordinates, or material coordinate and spatial coordinate. The material coordinate is the ink mark. Okay, this is like the cell number 10. Yeah. Not here. There's addition of cells just at the tip. So for this case, we can even disregard it, right? I mean, this thing is occurring due to elongation. Okay, in any case, the division is only at the very tip, because you're adding new material and it's flowing back, let's say. So here, basically, the idea is that you can look at growth like flow of fluids, right? So taking this Lagrangian or Eulerian coordinates is something that you take from hydrodynamics. It's two ways of looking at your system. So again, S0 tells me where the material is, okay, a specific cell, let's say, or a dot here is, and that's in red. And the spatial coordinate is basically the S, our median line. Okay, that's where it is in space, due to our reference frame. Okay. Now I want to show you just a bit more in details, in numbers, what I told you in words before about the elongation rate. So here you have, for example, a root instead of, an Arabidopsis root as opposed to the other root that you saw before. But it's nice because you can really see the single cells here. They're marked in yellow because you can see how, okay, here at the front, this is the apex. Okay, it's growing that way. This is the apex, and here you have 4 cells, if I remember correctly, that are added all the time. And then as as time passes, these cells, just one second, grow larger and larger and larger. You can really see the difference. Until here, they're all the same size. They stop growing. Yeah. What do you mean maximal distance of the root? I'm not sure I understand what you mean. Maximum size? Yeah. So you're asking whether it's if this is a universal thing for all plants? No, there's nothing universal in biology. So so you'll always have an area where you have where cells elongate, and they all stop at some point. Okay, how big this is, it changes between different plants. Within one species, it's relatively the same. Yes, I mean, apart from, of course, general variation, but yes, there's a characteristic growth zone or elongation zone for each plant. What you see below here, so you can decide relative to what. Here, this is v, the local velocity, which is the derivative of the spatial coordinate small s, right? Derivative with time. So it tells you basically how fast, if you put one of those ink dots on the root before, how fast it moves away. So this is relative to the apex. Okay, so it's as if the cells are moving backwards. Okay, of course they're not, right? But you have to choose relative to something. So here the velocity is 0 at the apex, and then it goes up to a maximum speed, and then it stays constant. Right, because it doesn't change anymore. When you're outside, this is basically the end of the elongation zone. And if I look at the derivative in space of this velocity, then you see that the elongation rate, E dot, is maximal somewhere around here, which is pretty much what we saw with the dots, right? We saw that there was a place in the middle where the distance between them is largest. And again, I have to stress that whatever is outside of this elongation zone, or growth zone, however you want to call it, whatever is here is fixed. It can no longer change, and no matter what stimuli you give it, light, gravity, whatever, it will not curve. It will not be able to change its shape. Okay? Super. So now we know that there is, what we have to do is basically, if we think about these coordinates, right? The material coordinate tells me what is the change, when I'm considering the change of curvature in time, the derivative in time, it tells me what is the change here on this cell, which is a local thing, right? Even with growth, locally, I won't see the difference. If the 9th cell is like between the 8th, 9th, and 10th, the curvature is somehow changing, I don't necessarily see a big change in curvature when they're growing, right? Because it's locally. But of course, the whole organ, we do see a change. So this means that we can no longer use the regular derivative, which is related to the specific black mark, okay? But we have to move to a material derivative, which is a co-moving derivative, okay? So basically, we mark this this is the material time derivative of kappa, and we mark it with a capital D, and it's just the regular derivative plus this is the I think it's called the convection term, right? So this is the speed at which you're moving. Okay. So now, if we go, remember that in the previous slide we had the derivative in time for kappa, but the regular derivative. Now we have to do some changes. So we had, this is our segment from before, a single segment. Now we change basically this derivative, the regular derivative, with the material derivative. Okay? So now this is at the level of the whole organ. Okay? So this is pretty nice because now we have the change of curvature in time as a function of blah, 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 blah. But this is the nice part, okay? Because now we have the growth rates at either side, locally, of course. I should have added the S and T. But this gives me now the full dynamics. But let's take it one step further, reordering things just to get a better understanding of this equation. So I can now rewrite this by making these definitions. So, oh, I didn't notice that it's all ones here. So the mean growth rate is just the average between epsilon 1 and epsilon 2, okay, obviously. Right? So I'm just averaging between the two. This is the average elongation rate. And there's a differential growth rate, which is the difference between them. So it's epsilon 1 minus epsilon 2, epsilon 1 plus epsilon 2. Sorry about that, okay? So the differential growth rate tells me how different are the two sides, the growth rates on the two sides. So if there is no differential growth rate, so the two sides have exactly the same epsilon, it's 0, right? But the mean growth rate will be just epsilon 1, let's say, okay? Assuming in this case, which I, by mistake, already did this case. So now I can, if I use these definitions, I can rewrite this equation in this form. And now I have basically the same equation, but in terms of something which I can understand in terms of the roles, the role that they have in this movement. So it's a function of the average growth rate and the differential growth rate. And for simplicity, for this, so this is the full equation. For our discussions in future, we can just take the approximation where kappa squared r is small compared to 1, okay? So the radius is, the radius of the thickness of our organ is significantly smaller than its curvature, okay? So this tells me basically that I'm looking at a very thin organ, okay? And in this case, I can just disregard this part here, and I get just the linear, this linear form. But this is very nice because this means that I can see, I'll just finish the sentence, that the change of curvature in time is basically a multiplication of two effects, the mean elongation and differential elongation, differential growth. So if either one of these is 0, although they're separate things to some extent, okay, I will not get a change in curvature. There was a question? Yeah. Actually, I think it should be just kappa r. It's just kappa. Yeah, there's no squared here. Sorry, this happens when you add a slide at the last minute. Okay, yeah.
It's cylindrical.
Yes.
So what do you mean?
So you can imagine we're here looking at everything only in 2D. Okay, this whole discussion is in 2D. We can also generalize it in 3D, but not in this discussion. But you can think just that, you know, 1/2 of the cylinder has one growth rate. Yes, it's obviously not like this obviously there's a gradient, right? It's not a clear cut this side and that side. But for the sake of this discussion, it's enough. Okay, so now with this equation, we can also extract important characteristic scales. So the length scale is r, the radius, and the time scale is 1 over E dot, 1 over the growth rate. So this is basically the time for doubling the length of a segment. Okay, so what can we do with this? Now that we have this equation, what can we say about it? Okay. up until now, if you remember, previously when we discussed all of this, the dynamics of these organs, they were as a response to some signal, right? So we were expressing the kappa dt, like the change of curvature in time, as a function of external and internal stimuli, right? Proprioception and gravity, let's say. Okay? In this whole discussion, I didn't talk about stimuli at all, right? The only thing that I did was connecting the kappa dt to the internal growth rates, right? So how do the two things connect? So let's look at this. We said that we don't have any shrinking, right? So both small epsilons are always larger than 0. So this means that the average growth rate is also positive, always, okay? So now, since we know that in our dynamics we have changes, right? Oscillations, we have changes in the direction of kappa, right? So it means that the sign of d kappa dt must change somehow. But we just said that E dot is always positive. So this means that the only thing that can cause us to change direction is basically the differential growth, okay? The sign of differential growth is what dictates the sign of d kappa dt. Why is this interesting? Why do we care about this? Because this tells us that delta is the only place where we can basically connect to our external signals, right? This is what is affected by the external signals. We also know this biologically, of course, right? Because we know that when we have a signal, we have the growth hormone, auxin, which is redistributed to one side. And this is what causes the fact that one side has a higher growth rate than the other. Okay, so it all makes sense. But also from a mathematical point of view, it's nice that we can identify this. So if we say the perception affects only delta, so if this is our delta, we say that this equals to what we had before on the one hand. On the other hand, we know that it is equal to this, right? The only thing that we have to do, though, is that we need to renormalize our beta and gamma in terms of dimensions, because here we have growth. We have R and E dot. So these are, with the tilde on top, is just the renormalized constants, okay? And now we're able to connect the dynamics, the change in curvature, in a growing system with the external stimuli, okay? And this stepping stone of delta is what allows us basically to play now with different things, okay? Here it's external stimuli, but I could plug in, instead of this, something else. For example, we have the internal mutations that I was mentioning yesterday, the other talk. So plants, even without any stimuli, you'll see that they tend to rotate a bit, but that's an internal thing. So what this means through this model is that I can say that now, if we're going to a three-dimensional picture, as you were asking before, now imagine that this differential growth, that one side grows at a higher rate than the other, rotates with time. Okay, the sides move. So if the higher growth rate moves in a circle around the circumference of the plant, it means that you also be rotating, right? Your growth direction also rotates. Okay, so we're assuming that there's no twisting. No, there's no twisting, because you're just constantly, first of all, this is a higher growth rate, so I'd move to the left. Now it's, okay, let's do it from behind. This is the higher growth rate, so I bend forward. But I don't bend, I grow in the direction of. There's no twisting. So basically, this is a very important point here, because although specifically here we're talking about tropisms, fine, so this is what I'm showing here, but you could think that if you want something else, an internal signal or some other complex signal, that's where you plug it in, okay? Okay, so now we have these two renormalized betas, which so I can again define, remember we had this B, the dimensionless number, which was a function of beta and gamma. So I now, instead of beta and gamma, I have beta tilde and gamma tilde. But this now, this is nice because now it can allow me also to compare, it's normalizing over the physiological differences of different plants, right? Because I have within here, I have the radius and the growth rate, apart from the sensitivities. So this is what allows me also to compare basically a tree with wheat coloptile. Okay? And I can also tell you that if I take the case of constant, what's called exponential growth, so this means that you have E dot is some constant everywhere. So if we saw before that real life E dot looks like this, the function of S, right? So what we're doing here is we're just making it a straight line, okay? Just for the sake of being able to give an analytic solution. And the analytic solution here for the steady state for this equation gives you this, okay? Which is a very ugly equation, but we have it. And in a second, I'll show you why it's good to have. So, okay, so I guess this is the most important slide of the whole talk today. How much time do I have? Great. Just 1 second. Okay, conditions for growth regulation. So now that we see that we're able to describe our growth, it turns out that it's not so obvious that you're still able to regulate your growth in such a nice way where you're straight at the end of the day. And we'll see that there are a few conditions that you have to meet. Okay, so I'll go through a few of them. One is passive orientation. So what this tells me, passive orientation means, let's say I start with some curved segment, okay? And now I start with a curved segment, and I say that the growth rate on both sides is identical. There is no differential growth, okay? Epsilon 1 equals epsilon 2. So what do we know from this equation? As we said before, if one of these is 0, so if there is no differential growth, even if we do have a mean elongation, okay, the kappa dt is 0. There is no change in curvature, which is fine. But it turns out that you have to take into consideration that even though you're not changing your curvature, what you are changing is the orientation, the angle, right? So if this is your segment, okay, let's say you started with this part, and now you grow with identical growth rates at both sides. You keep the same curvature, okay? But the angle, so if I look at the angle from this end compared to this end, it's a different angle if compared to if I look at the angle between this end and this end, okay? And this is what we call passive drift, passive orientation drift. And we can make a calculation for this, because now the question is, given that we know that there is this passive orientation drift, what do I have to require from the system so that you won't be affected by it? Okay, because otherwise this means that, imagine that you have basically a system that is passively, the angle is drifting just because of some problem, let's say, or due to something with the time scales. And we don't want to have this problem. So what is the requirement? So if I now define delta theta as the difference between the angle at S plus delta S, okay, minus theta S, okay, this is the difference in angle at the beginning and at the end of the segment. And this is equal to just kappa, okay, our curvature multiplied by the length of our segment, delta S. And if I substitute this in our equation and play with things a bit, what I get is this equation here, okay, which tells me how the difference in orientation changes over time, okay? And I see, okay, this part of the equation is 0, right? Because we have no differential growth. But so this means that we can now identify this term as a passive orientation term. And now it's obvious that in any system that we want, if we want to overcome this passive orientation, this term has to be bigger than this term. And if you make this requirement and you play a bit with the equation, you get that this boils down to having a gamma, which must be larger than 1, gamma tilde, the normalized one. And if you want to express this in a form that has meaning, you can rewrite this in this form. TC is the time for convergence, if you remember. Okay, it was the time that it takes for, if I put the experiment, do an experiment and I put the plant on the side, the time that it takes for the plant to reach steady state. Okay, so this tells me that the convergence time must be smaller than the average elongation time. Okay, because I want to converge before, so as not to fix my position. Okay, so this is one thing, and I'll just show you here, these are simulation. This is for a simulation with gamma smaller than 1, so the size of the curved zone is expanded by growth. Okay, this is just what we mentioned before, and the organ cannot regulate its posture, so it's constantly, it's trying to fix the mistake, basically, of this passive drift, and that's why you have these oscillations. Okay, even if we're using, let's say, betas and gammas that in the previous model, when there was no growth, they were fine. Okay, you could reach the steady state just fine, but because of this drift in this regime, it's not able to regulate its movement. Whilst here, you can see that it does it just fine. Okay. Okay. The second condition is avoiding fixation of curvature. What do I mean by this? I'll show you again this movie that we showed last time. And as you remember, the Cerebridopsis was overshooting. There were a lot of oscillations. Remember, we discussed about modes. So here it was, I think, second mode. It had two overshoots. But at the end, do you see the overshoot? No. And actually, it's a very good thing at that, right? Because if plants showed all of their constant overshoots when they're trying to fix some change of direction due to light or due to gravity, it would be a mess. It's not just because it's nice that they're straight, but it's important for physiological reasons, of course, right? Because you want the tree to be straight and not wriggly, so that it won't break up. So, but you could imagine that if you were to, we talked about growth zones, right? And someone asked here before about the size of the growth zone, whether that changes between plants. So it does. And you can imagine a case where if the growth zone is small enough, then while you're curving, you're in the midst of your dynamics to reach a steady state, but your material has already exited the growth zone, and you're stuck with that curvature now. And you're stuck with it forever. So that's a problem, okay? So basically, in order to make sure that you don't have this fixing of the curvature, what you have to require is that the time for the vertical element to be convected out of the growth zone. So the vertical element means we're starting from the side and we're going towards the vertical. So you're asking when the vertical part would exit, how much time it would take for it to leave the growth zone. So this is one time, and this is a convergence time from before. Okay? So you want to converge, right? So reach your steady state before you leave the growth zone. That's basically what we're saying. So here I didn't show how to get this, but you can, of course, read it in the paper. And when I now look at this relation, that I require that this be much bigger than this, I get this constraint on gamma. And again, I can rewrite it in this form. Where these are all things that I can measure experimentally, right? So they give me a bit of a feeling. So B, remember, B is a dimensionless number that relates shape with dynamics and tells me high values of B are the floppier plants, let's say, with low proprioception, and low values of B are like the wheat coloptile, where they're stiffer, so to speak, not mechanically stiff, but their movement is like this. And this is the size of the growth zone and your radius. Okay, so you see how the size of if the growth zone is very, very small, it means that you have a very strong requirement on B, on the type of dynamics that you can allow yourself. Is this clear? Yes, it's a function of many things, but in particular, it has also a function of beta and gamma, which are the sensitivities. Okay. And lastly, this is not really a condition for growth regulation, but this was an answer to a question that someone asked last time. When is it okay to use the equation from last time instead of using this thing, which is obviously more complicated, right? You have a lot more stuff going on when you take into consideration growth. So here what you can do is you take you can look at it at the end of the day, why use the other equation? It's from an experimental point of view, right? You do an experiment and you're asking yourself, can I use the simpler equation and just assume that L is fixed, or do I have to use this equation? So what you do basically is you can just compare the function for theta at the steady state of this model, the ACE. So ACE, I never said what it stands for, ACE and so on. It's angle curvature, and this is angle curvature E, elongation, okay? So the steady state for this model, which I showed before, and I compare it to the steady state of the regular AC model with no growth. And I ask myself, when will the difference between these two be smaller than some number, which is just the error in my measurements, let's say. Okay, so I arbitrarily chose this number, assuming that that would be in my measurement. And then when I play around with these numbers, with this requirement, I get that gamma tilde has to be larger than 6.2, okay? So this 6.2 is completely defined by my error bar, the error that I have in my experimental system. And then I get, again, if I rewrite this in a way that is tells me something from experimental numbers, I get that B has to be smaller than this relation here. And remember that, again, this 6.2 is something that changes according to your experiment. Okay, so now that I've thrown at you various conditions, I want to show some experimental validation for these conditions. So, I don't know if validation is the word, but to see how plants fit within these requirements. So here they did basically a variety of experiments over, as you see here, many different types of plants. And these lines represent, so the green line is, okay, let's start with the red line. The red line is this. So this is the condition for avoiding fixation. You have B as a function of the growth zone divided by R, okay, this here. So this is the square root, and it's this requirement. And the green line is this. So this is the condition for the condition for allowing you to neglect growth. So you see that all of the measurements are below this, so they all fulfill this requirement where you avoid fixation of the curvature, which makes sense because we don't see plants that have all these oscillations in their growth. They're pretty straight. And most of the plants also fit the requirement, this requirement. So for most of these experiments, I can, in fact, just use the simpler model, where I assume when I can neglect growth and just look at the steady state, and that will give me all the information I need. But you see that there are some outliers here, okay? And of course, the first condition of passive growth, there, if you remember, gamma tilde had to be larger than 1. But if here we require that gamma tilde is larger than 6.2, then obviously it also satisfies larger than 1. Okay? So we see that the two conditions that had to be met for regulation, both passive drift and avoiding fixation of curvature, are met in plants. Okay? And the third one is just for experimental issues or for to have an easier life. Okay, this is perfect timing. Now I want to, after I've filled you a bit with all these models and geometry. Okay, we know how to describe the kinematics of a plant. Yeah. Yes. Yes, but there, I mean, so on the side of the equations, yes, just the mechanism is different. So if here the differential growth in the wheat and Arabidopsis that I was showing was elongation of cells, that's the growth rate. With trees, it's something that happens to the wood. So there's either compression wood or tension wood, and I think it depends on which tree you're looking at. So either one side will contract or it will expand, and that's the differential growth that you get. The equations fit anyway. In fact, if you remember from the last talk, I showed 3 different examples of experimental data. It was for wheat, for Arabidopsis, and for a young poplar tree, 2 meters high. So even though the mechanism is completely different, the equations work just the same, because at the end of the day, we're just looking at a difference in rates. Okay, so now that we have all this, yeah.
Are they, do the cells are genetically different over different distances, so that, for example, they're more differentiated in one side and...
Okay, differentiation in roots, I can tell you. They're actually not differentiated through the growth zone, and they only start to differentiate after that, because there's also a whole hydraulic mechanism. You have like tubes inside for water going up and down and nutrients and so on. So at the stage of the elongation zone or the growth zone, they're not there yet. Everything happens after that. For the shoot, I honestly don't know. Actually, I have to check that. Another story. That's it. So I want to thank all the students in my group who are great and make it fun every day, and funding, and of course, all the collaborators. And thank you very much if you have any questions.